All right, so now we're going to take a look at the duodenum and the accessory organs. So we have already chewed up the food, we have masticated, we moved it around with our tongue and chewed it up with our teeth. We have swallowed it, it went down our esophagus into our stomach, and now it's going to leave the stomach. That chyme is now going to leave the stomach. Let's take a look at what happens there. We're going to use our mannequin here to help us with that. So what I'm going to do is I'm going to take out the liver here. We're going to come back to the liver, but right now we're going to take a look at the stomach. And so what's going to happen is we're going to get to the point where the food is now leaving the pyloric sphincter. Let's take a look at that here. So again, digestion is still going to continue, all right, and is completed in the upper small intestine area. So that's this area up in here, okay? This is the duodenum right here. This little part right here. Doesn't seem like much when you compare it to everything else, but a lot is happening right here. This is what we're going to focus on right now. Okay. Now with the pancreas and the gallbladder and the liver, those accessory organs, that's how digestion is going to continue and come to completion in our body. So the first portion here, like I said, the duodenum. The bile and the pancreatic juices are going to empty in there. And again, that's this. If we take a look at this picture, it's this. It kind of goes behind this section right here. Okay. It's also important in the digestion of fats because so far we haven't done any digestion of fats. Right? We've only talked about carbohydrates and proteins, so we've got to get to fats as well. Then digestion is going to end here as well. So after this point, it's all becoming, we're going to see absorption occurring. The body's going to start absorbing the nutrients and then absorbing whatever water is left over as well. So let's talk about those fats. There's three types of fats: neutral fats, fats and oils, phospholipids, and steroids. Now the neutral fats, those are coming from your plants and animals. The phospholipids help make up our cell membranes, and steroids are steroids. Now lipids are also non-polar, all right? So they don't have a charge to them. Water does have a charge to it. Remember, water is a polar molecule. The oxygen side is slightly negative, the hydrogen side is slightly positive. Now, because of that, water is and oil will not mix. All right? This is what we mean by insoluble here. We can see that we have the water here and we have the oil sitting on top. Now, lipids, okay, they will dissolve in alcohol. So if I had alcohol in here, we wouldn't have the separate layers. We would see the dissolving occurring there. And right about where that red tape is where the line is, the separation of the oil and the water. So some of the functions, right? So we use fat, we need fat, okay? But some of the other functions that we have for it is that, one, it stores energy. It's long-term storage. And it can store twice as much energy than carbs, all right? And now we can also use it for padding as well, protection of our organs, so moving around, jumping around. And then there's also insulation to help with homeostasis, control body temp, keep our bodies at a certain temperature there. And then transport, so transport of vitamins in blood. So again, the blood is water-based, right? And so the fat, the lipids, they're not very soluble. And so what we can do is we can use those to help then transport the vitamins along through the blood. Okay, so let's look at a basic understanding structure of fats here. One, fats actually have two monomers, the glycerol and the fatty acids, and they come together there like we see in this basic outline here. Now, fats, all right, your animal fats, our animal lipids, we call it fats, and then the lipids that we get from plants, we usually call the oils there. And then they makes up our adipose tissue that we learned about in Unit 2 with the histology as well. It also comes in two forms, saturated and unsaturated. And these become huge in conversations with diet because one is better than the other, the saturated and the unsaturated. And in fact, the unsaturated seems to have better health benefits than the saturated. And we'll get into why here in a moment. So let's take a look at saturated fats. One, they are solid at room temperature, all right? Animal fats. You take your bacon fat, you can leave it out. It'll get mushy, but it'll still stay solid, all right? You can stick your finger in it, but it'll still be solid. It won't be a liquid like the canola oil that you might have underneath the sink that's used for cooking, or olive oil. Those are liquids, right? And those are the unsaturated fats. So what makes a saturated fat comes down to the bonding. The hydrogens that we see on this chain here, every carbon has a hydrogen exactly all around it. So every bond here is covered on the carbon, right? So here's a bond to another carbon, to another carbon, to another carbon. And then we have a bond to a hydrogen here, here, and here. So the carbon is completely saturated, all right? And that's what we mean by saturated fats there, because it is completely saturated with hydrogens all around it. There's no carbon here that is missing a hydrogen. So looking at some health relationships here. So absorbed by LDL. So we have LDL and HDL cholesterol. And again, one is better than the other. And LDLs are, when you get your cholesterol screening, you actually want to have lower LDL numbers than HDL numbers. And it comes down to LDLs and HDLs are lipoproteins. They're going to transport the lipids around the body. What LDL does is it actually takes fats, lipids, and will bring them to the cells. And this is going to build up the plaque. And the plaque makes the blood vessels very rigid and narrow, so not very much blood can get through like we see here, and can actually cause blood vessels to start to crack. All right, now this can lead to type 2 diabetes and heart disease as well. And we call this atherosclerosis. Some properties of the unsaturated fats: they are liquid at room temperature, like I mentioned before, and these are absorbed by HDLs. Right? So what HDLs will do, HDLs will take those lipids out of the cell, off the cell walls, cell membranes, away from building up that plaque in the arteries, and will take them to the liver where it can be broken down more. All right. Now, like I said here, it may reduce arterial plaque as well. So let's take a look at these bonds again. So with unsaturated fats, we have this little kink that occurs, and that little kink is what makes it unsaturated. And that kink comes from a double bond. All right. So that double bond that we see here, and here, and here, and here, and here, and here. All right. So we have mono unsaturated. So there's one double bond. We have then polyunsaturated, where there are more than one double bond there. And that double bond, since it's there, then hydrogen can't bond to it with any of those carbons. So that's why it's unsaturated. We don't have as many hydrogens and not fully saturated around that chain of carbons there. So how do we digest fats? Well, that's where the liver comes in. And the function of the liver, there are tons of them here. Okay. So for instance, production of the bile. And the bile will then emulsify the lipids. It will break it down from big globs down to smaller pieces, which we'll get to and talk to later here. And then also it will remove extra glucose from the blood, stored as glycogen, stores iron and some vitamins as well, detoxifies the blood, which many people already know coming into this class, and also removes and breaks down bacteria and old red blood cells as well. So you can see the liver is a very, very important organ. And so that's why we get in trouble when we start having our livers start to break down on us. So let's talk about that bile that the liver makes. Now that bile is made inside the liver, and it's stored in the gallbladder here, this little green sac. It's stored in there. And what's going to happen is that when our body needs it, it will then be released, and we'll talk about how it gets released and where, but it'll get released, and it will start to break down those lipids that we've been talking about. And now when they break it down from the big globs here down to the smaller ones, then there's more surface area for enzyme action to occur. And then the lipase, which is the type of enzyme, lipase, ASC enzyme, that will then come in and digest the lipids down. All right, so the different ducts here. All right, so this is where we have to take a look at where the bile comes out of the liver, where it's stored, and then how then it gets into the duodenum. So the liver's hepatic duct will go ahead and will connect to what's known as the common bile duct. From there, the bile can then be stored into the gallbladder cystic duct. And then when the body needs the bile, it can then be released from both the gallbladder cystic duct and the liver's hepatic duct into the common bile duct into the duodenum. And that's what we're going to take a look at right here. Here's the common duct right here. Here's the liver's hepatic duct, and here's the gallbladder, the cystic duct here. And you see they come together, the hepatic and the cystic, into the common duct, which then goes behind the stomach and will meet and make its way into the side here of the duodenum. What about the pancreas? I mentioned before that the pancreas is an accessory organ as well. We have the exocrine gland and the endocrine gland. Now we've talked about the endocrine system a bit, and that's where the endocrine gland comes in to play. Now, endocrine produces insulin. The endocrine gland will produce insulin, which will control that glucose in our blood, right? And tell our body to start uptaking the glucose out of our bloodstream. And again, if you have diabetes, then your body doesn't do this very well. Now, the exocrine gland will create many enzymes that continue digestion of carbs, proteins, and lipids. It creates a lot of different enzymes, and we're going to cover a few of them here that you will be responsible, the ones coming up here, but we're not going to cover all of them in there. So, the pancreas here, we can see how it connects. Now, this right here is that common bile duct, right? And then the pancreas will connect as well and will secrete its enzymes into the duodenum here too. So, we're going to create enzymes and buffer in what's known as Acessory cells. From there, they will go through the pancreatic duct, into the common bile duct, and then into the duodenum. So, pancreatic enzymes that you will need to know, I said you're going to need to know some of them. Carbohydrates, pancreatic amylase. All right, so in our amylase CR lab that we did, and we talked about how we had those three solutions, one from the mouth, one from the stomach, and one from the duodenum. Well, the amylase in the duodenum is coming from the pancreas there. We have proteins that will be broken down by protease and trypsin. We'll have lipids broken down by lipase. And then we will have nucleic acids, RNA and DNA, broken down by nuclease. And you can see that all of these end with ASE. That is our suffix for enzymes. Now, enzymes are released as inactive forms within the pancreas, and then once the environments are correct for it, they will be activated just like we talked about in the stomach with the pepsinogen, and then being activated into the pepsin. So, all this digestion has started, all right? And the upper section of the small intestines, that's where you're going to receive the enzymes from the common bile duct, coming from the liver and from the pancreas. Now, inside the small intestines, we have a series of tiny fingers that increase the area of the tissue that is in contact with the partially with digested food. So the food actually will start to fall in to these spaces here, all right? And these bumps and these fingers here are called villi. Now, we talked about microvilli in our Unit 2 histology chapter, but these are a little bit bigger, right? So these are called villi. And you can see if this is the small intestine, you can see they are much bigger, right? Much bigger because microvilli are on the cellular scale here, and these aren't down on the cellular scale. And so this increases surface area, which allows for more absorption, and these are filled with blood vessels to carry away those nutrients into the body. So like I said, the microvilli, they're just like the brush, the border here of our little bumps and fingers here. And each villi, they can make digestive enzymes as well, and they will complete digestion too. And again, here we get a better view of where the food would fall into, so we have that further surface area going on there. All right, so we covered the duodenum basically right there, and the small intestines. And in our next segment of notes, we're going to take a look at the absorption as